Welcome to this lesson on complementary base pairing. Today, we'll explore one of the most fundamental concepts in molecular biology, how DNA bases pair with each other through hydrogen bonding. We'll specifically focus on adenine pairing with thymine and guanine pairing with cytosine. Understanding this concept is crucial for grasping how DNA maintains its structure and how genetic information is accurately copied during replication. DNA is composed of 4 nitrogenous bases. These are adenine, thymine, guanine, and cytosine. We often abbreviate these as A, T, G, and C respectively. Adenine and guanine belong to a chemical group called purines, which have a double-ring structure. Thymine and cytosine are pyrimidines, which have a single-ring structure. These 4 bases are the alphabet of our genetic code, and the way they pair with each other is absolutely essential for life. There's a specific rule for how DNA bases pair together, discovered by scientists Chargaff, Watson, and Crick. Adenine always pairs with thymine, and guanine always pairs with cytosine. This is not random. The pairing follows what we call complementary base pairing rules. If one strand of DNA has an adenine, the opposite strand must have a thymine at that position. Similarly, if one strand has guanine, the other strand must have cytosine. This complementary nature ensures that DNA strands fit together perfectly. The bases are held together by special weak chemical attractions called hydrogen bonds. A hydrogen bond forms when a hydrogen atom, which has a slight positive charge, is attracted to an electronegative atom like nitrogen or oxygen, which has a slight negative charge. These bonds are much weaker than the covalent bonds that hold atoms together within a molecule. This weakness is actually important because it allows DNA strands to separate when needed, such as during replication or transcription. The number of hydrogen bonds differs between the 2 types of base pairs. Adenine and thymine form 2 hydrogen bonds with each other. However, guanine and cytosine form 3 hydrogen bonds. This difference in the number of bonds has important implications for DNA stability. The single dash between A and T represents the 2 bonds, while the triple line between G and C symbolizes the 3 bonds connecting them. Because guanine-cytosine pairs have 3 hydrogen bonds compared to the 2 bonds in adenine-thymine pairs, GC pairs are significantly more stable and stronger. This means DNA regions with a higher percentage of GC pairs require more energy to separate the 2 strands. This difference in stability affects various biological processes, including the melting temperature of DNA and the ease with which strands can be separated for replication or transcription. Complementary base pairing serves several critical functions in biology. First, it ensures accurate DNA replication. When DNA copies itself, each base on the template strand specifies exactly which base should be added to the new strand. Second, it maintains the double helix structure of DNA, keeping the 2 strands at a consistent distance from each other. Third, it enables complementary strand synthesis, allowing cells to create a complete copy of the genetic information whenever a cell divides. Let's review the key points from this lesson. First, adenine pairs with thymine using 2 hydrogen bonds. Second, guanine pairs with cytosine using 3 hydrogen bonds. Third, GC pairs are stronger and more stable than a T pairs because of the extra hydrogen bond. Finally, this base pairing system is essential for maintaining DNA structure and enabling accurate replication of genetic information. This elegant system ensures that genetic information is preserved and transmitted faithfully from one generation to the next.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.